All right, then Venkat, your co-host, okay? So,
yes, Professor.
Yeah. Good evening, all of you. It is today we are on this 9th edition of this discussion meeting. We have 4 speakers as usual. The first speaker today is Professor P. Venkatakrishnan, and he is going to talk to us on building bridges and forging bonds for functional organics. As you know, I will give a warning after 20 minutes, and then in 5 minutes you'll have to close. Then we'll have 5 minutes for questions. Thank you, Venkat. Let's get started.
Good evening, all of you. Hope you're able to see my screen, and
Yes.
I hope I'm audible as well.
You're audible. The screen is visible.
Yeah.
It's great. Yeah, thank you.
Thank you, Professor Pradeep, for having given me an opportunity to share some of the research work in this experiment theory interface initiative that is started by you. Thank you very much. So let me share some of the research work that has been happening in our laboratory for the past 6 to 7 years. Of course, I will be picking few of them and then presenting it to you. Okay. So as the title says, it is building bridges and forging bonds. So obviously you would expect 2 sections. One is about forging bonds and another one is about building bridges. So I will take you through the slides and then describe the aspects that we are going to deal with. Good. Yeah, so this slide gives a very brief introduction about our activities at IITM. We are basically organic chemists. What we do is we take the expertise from organic synthesis and use the knowledge from the physical aspects of organic chemistry to build functional organic materials. Okay, so basically to summarize, we have simple reactions to construct carbon-carbon, carbon-nitrogen, and carbon-oxygen bond-forming reactions. Through this, we would build 1-dimensional, 2-dimensional, and 3-dimensional organic architectures and derive information about their characteristics, be it photophysical properties or thermal properties or electrochemical properties, redox, and so on. Then based on that, we would test their potential in organic electronics. For example, transistors, solar cells, sensors, and organic LEDs are some of the areas that we are working on. And being an organic chemist, we would always be interested in developing some of the reactions. So developing novel methodologies to construct various set of molecules. And recently we have ventured into the macrocyclic chemistry, whereby you create cyclic molecules and look at their molecular recognition potential. Okay, so I will give you some of these aspects in this presentation, basically. So I would say the present era is full of polycyclic aromatic hydrocarbons. Every issue of American Chemical Society or Wiley, for example, you will definitely see some of the aspects of polycyclic aromatic hydrocarbons discussed. This slide summarizes the overview of different types of polycyclic aromatic hydrocarbons. These days, they are being called as nanographenes, particularly because of their dimensions. And one can create such kind of structures with the organic synthetic expertise. Okay, so they are atomically precise. So you can create long 1-dimensional graphene sheets. You can do the functionalization at the edge. You can also functionalize them at the core. You can also deform them, the graphene sheets, basically, by introducing a larger sized ring, such as 8-membered ring or a 5-membered ring at the core or somewhere between the graphenic skeletons. You can also dope them with heteroatoms as is shown here. Okay, so all of them are expected to induce different properties or bring in interesting or exciting properties to these graphenic structures, and hence they are of great attention these days. Very importantly, how do they construct such kind of large graphenic structures? That could be the question that is right now lingering in your minds. And what I would say, what I would tell you is the Scholl reaction is mainly the reaction that is responsible or that is being used to build such larger, well-defined polycyclic aromatic hydrocarbons. It is one of the simplest and oldest carbon-carbon bond forming reactions, I would say. What basically it does is it's just coupling two aromatic units. For example, here two aromatic phenyl units are connected via this carbon-carbon bonds. Likewise, you can start from a simple benzene, build a super benzene, and then make a larger graphene of carbon 222 unit using such Scholl reactions. Okay. Other than the desired reaction that you would want to have whenever you are doing Scholl reactions, you often end up with a lot of surprises. Okay, that is particularly coming from the competing side reactions that one would often see whenever somebody works on Scholl reactions. They are like rearrangement reactions, for example. Bond migrations could happen, and hence you may not expect to see the product that you would desire to obtain. Okay, so rearrangement reactions are the main culprit. You also have the oligomerization as a competing reaction in these conditions, because you're talking about oxidative coupling. You can promote carbon-carbon bond formation in multiple ways, and hence they would lead to polymeric structures. So this is another competing reaction, you would say. And other than that, the over-oxidation products and so on are competing side reactions. There is no systematic understanding as to how one would go about designing a structure to get the structure what you actually want. Okay? So there is a lot of research interest in this area. We are trying to understand what is actually happening in this kind of reactions. Okay? So I'll give you a brief idea. Mechanistically speaking, this Scholl reaction can happen in two different ways. And both the reactions that are proposed here, such as Adrenium cation mechanism and the radical cation mechanism, are commonly accepted. And when you look at the Adrenium cation mechanism, if you consider such a very simple compound whereby you are looking at particularly the carbon-carbon bond formation between these two arenes, you take this compound, subject it to Scholl reaction conditions. What happens is first the protonation, thereby you create an Adrenium cation. This Adrenium cation in a Friedel-Crafts manner forms a carbon-carbon bond, and afterwards loss of two protons and then two electrons results in a desired cyclic compound. Similarly, when you look at the other reaction mechanism, which is the radical cation mechanism, the first step seems to be the oxidation, and that is often seen with an electron-rich aromatic such as this denoted here. So you have an oxidation, you create a radical cation, and this radical cation once again forms a Friedel-Crafts type reaction, forming a carbon-carbon bond, resulting in a distonic cation radical. So after removal of an electron, you create the dicationic species, and loss of two protons results in an aromatic species. Okay. So what you see here, there is an Adrenium cation, there is a radical cation, but in between somewhere you also generate a dication. Okay, so all these species are known to coexist in the medium, and hence a clarity about the reaction mechanism is not there. At that point of time, we would depend reliably on the calculation aspect. So theoretical calculation helps to arrive at a meaningful explanation as to whether this follows an Adrenium cation or a radical cation. So most often you would find these mechanistic proposals supported by calculations in the literature. Well, so we have gotten some idea, basic idea about the Scholl reaction, and I will describe you taking few skeletons and tell you how we have employed the Scholl reaction to construct carbon-carbon bonds or to build a smaller nanographene in the laboratory. So first example that I'm going to tell you is about fluorine. Okay, so fluorine is a very small molecule, as you see. The interest on fluorine is mainly coming from the success with polyfluorines in organic LEDs. So fluorine as it's a very small skeleton, so it has a defined conjugation length, and hence absorption and emission is mostly in the ultraviolet region. So to expand the pi conjugated length, one can use Scholl reactions. and thereby you have an extended conjugation. You can in principle shift the absorption and emission to the visible region. So keeping this in mind, we ventured into this, made various molecules, both two-dimensional as well as the three-dimensional skeletons as is shown here. They all exhibit yellow color, which means they have absorption in this visible region. And they are all blue emissive as is shown here. They have a very high fluorescence quantum yield of up to 97%. Okay. So now, because you have very high fluorescence quantum yield, any change, minute change in the emission intensities could be very informative, particularly if you are looking at the sensing aspects, for example. Being an electron-rich skeleton, these fluorines could be ideal candidates to sense electron-deficient aromatics, and hence our attention was more into electron-deficient nitroaromatics, which are otherwise popular explosives. Okay, so we use them, and these compounds are reliably sensing these nitroaromatics to a concentration of up to 0.2 nanogram per ml. Not only in solution, but they have also shown to detect the vapors in thin films and also on solid substrates. Okay, so when you expose the nitroaromatic vapors to this pi expanded fluorines, you would see them being sensed nitroaromatics. Okay, so this is one of the aspects. And what more we can have with such large pi aromatics is the self-assembly aspect. These molecules, when you put them in solution, upon increasing the concentration, one would find that they have a very nice self-organization. Both in the film state and also in solution, one could characterize their self-assembly aspects. And luckily, we could grow crystals of them. And in solid state, we find that these compounds stack one on top of each other, establishing a dimeric phase-to-phase assembly. And hence, such kind of organization are very beneficial in terms of extracting the charge transport behavior in such molecules. Keeping that aspect in mind, with the help of Professor Soumya Datta from Electrical Engineering Department, we could fabricate the field effect transistor devices wherein the organic semiconductors are used. These molecules are serving as the organic semiconductors. These are put on a flexible substrates. Right, so you can have flexible electronics, and we have demonstrated the mobility being as high as 10 to the power minus 3. These mobilities are quite stable actually, and these molecules are quite stable. One can store the devices for a period of 1 year, and you would see nearly no decay in their performance. Okay, so in that sense, these are wonderful materials. Now, another molecule I will take and then talk about how we went about manifesting this Scholl reaction onto these skeletons. Here is a similar analogous compound. Okay, it's called carbazole. You have nitrogen in between the two arene rings, not the carbon as in the earlier case. To take this carbazole and put it into the Scholl reaction conditions, what you find is that it would quickly undergo dimerization to establish the carbon-carbon bond, thereby creating a bicarbazole. Okay, so we have tested this for various substrates, and you would often see the carbon-carbon bond formation established between the two carbozoles. We have understood the mechanism. In these cases, it follows via the radical cation pathway, as I described earlier. Keep that aside. Now, these compounds, okay, so you have these dimers of these carbozoles resulting in bicarbozoles. They do have a small deviation from planarity because of the repulsive interactions faced by these two ortho hydrogens of these bicarbozoles, and hence the planes of these molecules are no longer coplanar. Okay, so they are twisted to an extent of about 24 degrees or so. And hence what we thought was these compounds have better redox properties, they have good absorption and emission properties, and hence why can't we replace the expensive spiro-OMe tag and then test its whole transporting material in perovskite solars. So we made few basic skeletons of bicarbozoles, and then we tested their whole transporting ability when putting it onto the perovskite solar cell configuration. What we find is these small molecules could indeed perform better. You don't need to have an expensive spiro-OMe TAD as a whole transporting material, but you can synthesize simply these compounds and then put them into the perovskite device configurations. they would withstand a stability of about 1000 hours. And this is demonstrated recently in collaboration with Professor Jay Chang from Taiwan. Now I discussed about two aspects. One is taking fluorine, how do you go about elaborating? Then we talked about carbozoles, and then how we go about elaborating via Schall reactions. You can even elaborate the skeleton further. The bicarbazole that I talked to you earlier can be further dimerized to give you a tetrameric skeleton using the same Schall reaction. Just imagine how complicated it can grow. Okay, so you just take the dimer, you can make a cyclic tetramer. This is demonstrated in the laboratory. You can also think of annulating the nitrogen fused aromatic onto a superbenzene. Okay, so thereby you retain a planarity, but then you introduce the properties that are arising from this heteroatom onto such large aromatics. You can also fuse them further, thereby you create a ovalene type of structure which is doped with a nitrogen, and hence the photophysical as well as the electronic properties are expected to vary for this compound. One can think of exciting photophysical properties arising from these materials. Similarly, we have also constructed triangulates resulting from the simple carbazole unit based on the understanding that we had here, that we had developed earlier. One of the positions of the carbazole is often blocked in order to minimize the side reactions. That's the only objective. And once you do that, you are able to get a very well defined graphenic structures in appreciable yields. Now, although earlier examples were completely planar compounds, right? So we have also been able to twist the carbazole molecule by doing a bifunctionalization at both the ends of the carbazole. For example, you take the arene unit, these two arene units, anchor it onto two ends of the carbazole. You would try to cyclize them to form a helicene, and this helicene once again undergoes a Schall reaction to give you a circulin. So now what you've basically done is you've twisted the carbazole to form a helicene, and you've connected it further to introduce to create a more strained molecule, which are otherwise called as semicirculins. And the twist angle and the twisting nature of these graphenes would create a defect onto a larger graphene skeleton in case if you are planning to further elaborate them. And all these chiral aspects, because of the twisting, can be introduced into this structure. Okay. Recently, there are reports with such twisted molecules to create chiral light from the organic LED devices. And elaborating the same concept further onto coumarins. Coumarins are popular bio-friendly skeletons. They often have restricted conjugation, and thereby they limit their emission into blue region. But if you say that you are expanding the coumarin, then you will be able to push the emission and the absorption into the visible region. Okay, keeping that aspect in mind, we started to build the skeleton appropriately and then accomplish Schall reaction onto this coumarin. We could achieve starting from blue to yellow and all the way up to red by simply fine-tuning the functional groups and simply tuning the design so as to achieve a large polycyclic aromatic hydrocarbon embedded onto coumarin action. So you could see the solutions are emissive, and also the solids, they are luminescent, and one could construct such kind of molecules using the Schall reaction strategy. Well, till now we have talked about the forging of bonds because we have connected the two arene units via the carbon-carbon bond. Now I am quickly going to summarize you about the bridging of the molecules. Okay, so let's talk about macrocyclic chemistry. This is one of the areas that we had started working on recently. It is dated back to 1972. This chemistry is not a new chemistry. It is dated back to 1872, and in only 1970s it has started to mature. Okay, from the chemistry of the Nobel trio, Jean-Marie Len, Donald J. Cram, and Pedersen, when they shared the Nobel Prize, from that time onwards, this chemistry has evolved drastically. Depending on the arene units, you would name them as calyxarenes, resorcinarenes, pillarenes, and tetramethoxy resorcinarenes, and so on. What you see basically here is 4 of the aromatic units are connected via the bridges. Bridges here are nothing but methylene or methane bridges. When you connect them, these arene units are like in a bowl shape, as you are seeing here, so that a molecular recognition aspect can be achieved. Later on, after this chemistry, one could build this kind of tub-shaped molecule by replacing such arenes via a heteroaromatic unit. So they are called heterocalyx arenes. You have various kind of heterocalyx arenes that are reported in the literature. The main drawback in such kind of macrocyclic unit is they are not showing exciting photophysical properties. If you want to make use of the fluorescence properties and then monitor the recognition phenomena, you may have to attach the fluorophoric units onto the backbone of the macrocycle. Okay, what we thought was it is a very cumbersome process. Macrocyclic synthesis itself is tedious, and then attaching a fluorophore onto it would be even more cumbersome, resulting often in poor yields actually. What we thought was we can replace these building blocks that you have, okay, the arene walls, by fluorophoric arene walls, and hence you can construct a macrocycle that is overall fluorescent in nature. So the immediate thought process that came to our mind is coumarin. Coumarin, you know, as we have been working on coumarins, these are also brightly luminescent compounds. They have a variety of applications. And one more advantage with coumarin is you can basically prepare it from the starting material, such as phenols. Okay, a hydroxy compound, you will be able to easily do a condensation reactions intramolecularly to get such coumarin fluorophore units incorporated. So this was running at the back of our mind. So we started off with this skeleton, and we ended up with this, and we called it as coumarin 4 arenes, or call it as ring of coumarins. Okay, so this could be made. Variety of coumarin 4 arenes were made in the laboratory with reasonably good yields. And the exciting property with this coumarin is when you have this substituent at this site, okay, if you change the variation here, what we found was you will be able to rigidify the skeleton. Right now, the substituent gives its flexibility, and hence it remains in one particular conformation in the solid state. This is called a boat conformation, whereby these two arms are lifted up and the other two opposite arms are flat. Okay, and as we
4 minutes left.
Thank you, Professor. Yeah, I'll be finishing. So this molecule as such is fluorescent. It is blue emissive, as you see under the ultraviolet light, and it is also colorless. In solution state, as expected, this molecule exhibited a nice flip-flop motion, and hence you see a conformational switching which can be monitored when you lower down the temperature up to -90 degrees. So at room temperature, they are very fast in equilibrium between this form and this form. They are in rapid equilibration, and hence the NMR technique is not able to distinguish it. But when you lower down the temperature, this process is slowed down, and hence you are able to distinguish them. And the averaged peak is what you see at the room temperature. So basically, from the board form of this to the board form of this, via this form is what you would see under room temperature conditions. Okay, so this conformational switching aspect is quite advantageous in a sense that you will be able to put in various molecules into the cavity of the macrocycle and then look at their molecular recognition. And we tried various guesses here, and what we find is mainly the ammonium cations are being selectively captured by these macrocycles in a ratio of 2:1. Okay, so you have two host molecules capturing the guest, which is often the ammonium ion. Well, so can do the host guest chemistry. We thought of rigidifying the conformation. So for that, we need to use rigid rotors here. And the groups that are being used here are the isopropyl groups, which are bulky enough. And hence, this carbon-carbon bond is a little more rigid. And it doesn't allow it to undergo a fluxinal motion that you see in the earlier slide with the other substituent. Okay, so this isopropyl group keeps it more rigid. How can you confirm it? You can do again the variable temperature NMR experiments. What we found out was this compound seems to be rigid. If you make a coumarin from this starting material, once again this is rigid. You don't see the signals being split into two different conformations, which confirms once again that it is a rigid conformer. Keep in mind, such rigid skeletons with a defined void can sometimes be very, very specific and selective in terms of molecular recognition. So to our surprise, what we found was the same ammonium ion, when it was titrated with this macrocycle, what we found was the association between these two was very, very strong when compared to that of the flexible macrocycle. It's about 450 times stronger than that of the flexible molecule. The behavior of these rigid skeletons are quite similar if you compare a drug reaching a receptor site in our body. Okay, so it is quite strong. It is very, very selective and very specific. It can capture with our strong binding affinity when compared to that of the flexible one. Not only set ammonium ions, we have also examined fullerenes C60 and C70. C60 is a spherical molecule and C70 is kind of an oval shape, and it has a higher affinity towards C70 over C60, and the times is about 2.5 times stronger binding affinity with C70. So in principle, if you are able to increase the binding affinity of this macrocycle, you will be able to separate the C70 impurity with C70 mixture, or you can think of having otherwise. Okay, so this can be easily monitored by the fluorescence titrations as well. So here we had used the calculations aspect just to determine the strain energy. How much is the strain built into the macrocycle so that the conformational motions are arrested? We will see that upon increasing the bulkiness of the alkyl substituent, the strain energy is so high that it doesn't allow the conformational switching to occur. Okay, and that's one of the reasons why this molecule is quite rigid, and with an enormous strain energy, the conformation is quite intact, keeping the void with a defined size so that a selective guest capturing can be achieved. So with this I stop. I think I was able to give you a flavor of what we have been doing, and the take-home message is the molecules, if a priori, can be designed or calculated, or if a particular device configuration could be calculated and then proposed, probably will be able to take this research in a with a very jump forward. Thank you very much. Thank you very much. I thank all the funding agencies and my students, and thank you very much for your kind attention.
Okay, thank you. Thank you very much. We have just a few minutes for questions. Thank you for finishing reasonably in time. One question, of course, there is one question there in the chat box. Have a look at that and then probably respond just after this question. You talked about circularly polarized luminescence that's coming from this system. And I understand that that is also tunable, circularly polarized emission, right?
Yes, Professor.
Yeah, so these systems, if you are in a position to create such a circularly polarized thing by combining, you said that similar systems of this kind can also act as donor acceptors or host or guests or whatever. So, you know, you can probably create systems wherein circularly polarized luminescence can be combined with other properties. So is there some possibility of the so-called functionality that you have here, luminescence, circularly polarized luminescence, to be combined with additional properties such as, let us say you put with a nano system, create plasmonic circularly polarized materials. So you create magnetism, and along with this, so that creates a possibility of creating new functional properties with hybrid properties, right?
Yeah, right.
What is happening in that direction, or is there something that you are thinking of?
This is I think this technique, as you suggested, Professor, this is not yet blooming. I think only the helical molecules or the circulanes are being employed onto the devices such as organic LEDs to create circularly polarized light emission. Combination of properties, as you suggested, is a wonderful idea, where one can combine this plasmonic ones and see if that can be an enhancement in the G factor under the circularly polarized luminescence. That could be one of the things that can be examined, but I have not seen any reports.
You talked about these systems assembling to form structures.
Yes,
Larger
Professor.
structures. Now, these assemblies are also circularly polarized. So there is an inherent molecular sort of chirality. Assembly is also a chirality. Is this like that?
To achieve it in a very purest fashion, Professor. Right now, these molecules, if you see, they have both the enantiomers actually. So helical can be a positive way and in the negative way.
Right.
We have not yet separated the enantiomers. Once you separate them, then you will be able to have enantiopure. Yeah, right.
Thank you. I have other questions. Sujan is asking a question, and Deepak Patel, you have a question there on the chat box.
May I go ahead, sir?
Yeah, go ahead.
Yeah, please.
Yeah. So my question is like, is conformational switching or conformational rigidity, which you are talking about, only the result of steric effect, or is there any electronic effect also involved?
Electronic effect is not contributing here because because these two aromatic units that you have, okay, to form a bowl, they are spaced actually through a inert group. So you do not have an extended conjugation, I mean. Okay, so there is a break in conjugation. So if at all there is an electronic group, it is within the molecule. Within the molecule means within the unit that is constructing this macrocycle. So definitely the electronic effect is not playing a role here in rigidifying the conformation. But what is more important here is the steric effect that is given by the alkyl substituents at the bridges actually.
Okay, one more question is
let's move on. Let's move on because of time. Let's move to the last question. Deepak Patel, I see that question there.
Can we use this Shaw method to synthesize one-dimensional extended graphene ribbon like molecules with more defects and a terminal functionality, which can show better electronic properties? Yes, it is indeed possible, Deepak. But it all starts with the molecular engineering. So I think in principle, it is possible. It is there. I think there's one recent chemical science paper also on this actually.
All right. Thank you,
Thank
Dr.
you.
Krishnan, for this wonderful talk. And let's move on. The next speaker is Kamal Kishore. He's going to talk to us on seismic metamaterials to damp low-frequency surface disturbances. You can share your screen, and as usual, I will give you a warning after 20 minutes. We'll have to follow the time strictly. Thank you.
Yeah. Am I audible?
You're audible. Your screen is not visible yet.
Sir, can I like off the screen because there is a low width network issue?
Yeah, I mean, you want to speak without the screen or what? I mean, you can switch off your video. Yeah, sure.
Yeah, sure. I can. I'm sharing. Yeah, is it visible?
Yeah, it is. You'll have to put full screen though. Yeah. So please run.
Yeah. So hello everyone. My name is Kamal Kishore, and I'm a MS scholar in Mechanical Engineering Department. I'm working under Prabhu Raja Gopal sir. My research work is related to seismic metamaterial. So here today we are going to discuss how seismic metamaterial can damp low frequency surface disturbance. So this is the outline. We will discuss about how to design and numerical studies. So metamaterial is composite material that can be engineered to exhibit unique properties, and seismic metamaterial can be defined as can be designed to counter the adverse effect of seismic wave. So this low frequency to avoid this low frequency. Sorry. So yeah. So I'm sorry. So metamaterial is a composite media that can be designed, that can be engineered to exhibit unique properties, and the seismic metamaterial is designed to counter the adverse effect of seismic wave. So frequency, as this low frequency wave can cause structural damage as well as damage to sensitive equipment.
What do you mean by low frequency? How low is the frequency?
Yeah, so mainly seismic vibration causes generate frequency up to like 0 to 10 hertz. Now, there will be other frequency as well, but this low frequency will be very catastrophic, and they can damage structure breeze and other things. So that's why we are focusing on these low frequency vibrations.
Okay.
So over a period of time, researchers have done studies. So Sebastian proposed a Bragg scattering in 2013. Bragg scattering seismic metamaterial, where he performed experiment creating holes in substrate, soil substrate, and he obtained a narrow band gap around 50 hertz, which is way too high for seismic disturbance. So later on in 2016, Columbia investigated and found a tree as a naturally occurring seismic metamaterial. Again, this was effective in 30 to 40 hertz range, and which was not applicable for the seismic metamaterial. So
I'm sorry, Kamal. It is difficult to understand. What band gap are you referring to?
So band gap, I'll discuss later about band gap, what is band gap, and how it is going to change the seismic vibration.
Yeah, but when you say zero band gap, zero frequency band gap and all that, we understand band gap in terms of electronic materials. Okay, I don't understand what this is. So therefore, there is some lack of clarity in this. I'm sorry about that, but please, if you can tell me, it will be useful to hear your story better.
So there are like, we can attenuate the wave by Bragg scattering. So Bragg scattering is mainly, it is kind of a destructive interference. Like suppose there is there will be a two wave and the path difference will be like some path difference will be pi, and there will be some destructive interference which will cancel the energy, and it will shield it will stop the propagation of wave.
Yes. But what is the band gap coming?
When the wave will interact with our seismic metamaterial, so there will be destructive interference, and wave will attenuate, and either it will attenuate or it will reflect back. It will not pass through the metamaterial.
Yes, but why is it called a band gap?
Because it will be for some certain range of frequency, and it will stop some certain range of frequency and not below this or above this.
Okay, thank you.
Yeah. Later on, researchers have done studies for zero frequency band gap, where they have used clamping of metamaterials. Earlier researchers were doing just simple, they were putting some simple resonator in soil, and and they were studying about how much band gap is creating. So later on, few researchers have done studies on layered soil. Like earlier, all studies were based on homogeneous soil, which is not practically possible. And they have studied the effect of a layered soil on band gap. So, as we have discussed about research has been done over a period of time, but this study typically give us a very narrow band gap. Also, the resonator height is more than 10 meters. So the installation cost and practical feasibility is not possible. So our objective is to develop a low cost, locally resident, low cost, locally resident frequency stop band seismic material that can attenuate the wide range of low frequency. And also these resonator, these metamaterial can be used in all geographical conditions. So this is the proposed seismic metamaterial arrangement. As we can see that metamaterial can be installed around the building we are going to shield from seismic wave, and this seismic metamaterial can be installed in rectangular or square arrangement, but we are considering the annular ring because it will minimize the number of resonator used in a wide area. So next, so researcher have shown the band gap in different range of frequency. In 2016, Marco studied the cross shape and the circular resonator. The effect, the cross shape resonator have wider band gap, but it is difficult to manufacture. So also the height of Marco has taken the height around 10 meters. But again, this is not feasible to in practical implementation. So this is our proposed seismic material, where we have taken a square lattice with the diameter around 2.5 diameter around 1 meter and height is around 2.5 meter. This is a concrete bed where we have embedded around 0.5 meter inside the concrete bed, and this is a substrate, soil substrate, and we have performed eigenfrequency analysis to find the band gap. So we have taken two type of resonator. One has a height around 2.5 meter and other has a height 3 meter, and the diameter is around 0.5 meter. So these are material properties. We are using steel resonator and concrete bed, and the steel resonator is covered around by autoclave aerated concrete. So this is a phononic dispersion analysis. I have performed phononic dispersion analysis in console multiphysics. So I solve eigen equations and plotted that data in MATLAB. So we can see that there's a sound cone, and if wave is inside the sound cone, which is this region, so there will be the surface wave will be inside the sound cone. But since there is no surface wave inside the sound cone, we can say that we have a wide band gap of 0 to 23 in case of 2.5 in resonator 1. Same study is done for resonator 2, where the band gap is around 0.0 to 45 hertz. So to study the further the validate our study, we perform dynamic response analysis. This is our model where we are exciting up. We are exciting here, and we have installed two probes, one before the metamaterial and one after the metamaterial. We have taken an array of specific metamaterial. But perfectly matched layer is used to attenuate to avoid any reflection from the boundary of boundary. So we are using a Hanning window. Sorry, not this one. Okay. So we are sweeping a frequency range from 0.01 to 60 hertz. So this is a transmission plot where here we can see that in the simulation result that the wave is not passing through the seismic metamaterial, and some wave are also going inside the substrate. Our transmission plot also shows that we are getting quite high attenuation. So this is the same. This is for resonator 2. Again, the result is same. So moving forward. So this is the visualization animation. We can see that how it works. So this is with metamaterial, and we can see that wave is not passing our metamaterial. And here I haven't used any metamaterial, so I'll play this as well. So we can see that wave are continuously moving. There is no stoppage. Again, this is for second resonator, and effect is again we can see that wave is not passing our metamaterial. So further to investigate our study, we have performed time transient analysis. This is our model where I have taken a circular concrete bed, and it embedded. I have embedded my resonator inside the concrete, and the filler material I have used is autoclaved concrete. So these are simulation parameters. I have used a Hanning window as a excitation signal. So yeah, this is a 19 hertz Hanning window with the 19 cycle, and the FFT plot we can see that yeah. So this is the concrete bed where this where resonator is embedded inside, and the material is filled inside these remaining gaps by autoclave. Yeah. So this is our probe plot. We can see that the red plot shows the attenuated frequency, and the blue one is outside metamaterial, where we have measured at equidistance, and we can see that wave is attenuating. Also, we can see that in our simulation studies, that wave is not passing through our metamaterial. Again, I have increased the number of number of the resonator in so that to study like if there will be further increase in shielding. So earlier it was like around 85% reduction. When I increase the number of resonator, then the total reduction is around 90%. So I further increased the number of resonator, but result was almost constant. So I'll play the animation for like a better understanding how it is attenuating. Yeah, so we can see that wave is not passing through our metamaterial, and in all other direction, wave is going. Yeah, this is another an animation which more. Yeah, this is a prop plot for 5 pro metamaterial, and this is for 8. So the total energy reduction was around 85 to 90%, which is good. But we are trying to achieve around 95 to 98% reduction. So our future work will be to perform experiment and validate the numerical data. That's it.
Thank you, Kamal, for this presentation. Well, some things did not go through me, but nevertheless, I have this following question. So there are two aspects. Of course, you are in a position to shield low frequency modes, and that is useful for preserving sort of structures. But your waves are traveling longitudinally on the surface.
Yes, sir.
Now, you can also have, in a seismic situation, you have a transverse wave propagation from the interior. So I would like to know, how do you plan to attenuate that?
So, sir, my main focus is on surface wave, which usually travel in vertical and as well as horizontal direction. So the particle motion is always in circular, elliptical shape.
I understand, but you can also have, there are many seismic activities near, resulting in propagation. As a result, you have your materials thrown out. I mean, it's essentially a pressure, a wave propagating towards the surface. So that's a transverse motion.
Yeah. So for that, we are using concrete bed, I guess. That should sustain.
Okay. All right. So that is one thought or one kind of question that you have. I mean, I have. The other point is that the low frequency modes are attenuated very nicely, all that. The implementation of these will depend upon cost, isn't it? So can you please comment on the cost effectiveness in comparison to other methods that are, I don't know what other methods are used, but can you compare?
Yeah, so the major advantage of this is other method has we use other system in building, so that need proper maintenance, and there needs some continuous maintenance, and the cost is as much as high for this regenerator. So for our regenerator, the one more advantage is like we will be shielding our whole region.
Okay.
And it will be like at some distance shielding.
All right. Thank you. Thank you. Are there other questions, colleagues? Are there other questions? Sujan and Soumya have questions. Yeah.
Yeah. So I have one. I mean, like very fundamental questions. Like, what kind of what is the building block of this kind of material? I mean, because I didn't get any feel of those things. I mean, is available, or how we'll build this kind of material?
Sorry, can you come again? I didn't get.
These structures are constructed out of what? I mean, these are steel and concrete.
Yeah,
That's
yeah.
I understand.
So this is just a periodic arrangement in such a way that it is creating some. Yeah. It is creating some path difference, which is creating some destructive interference.
So this is basically to avoid any penetration of waves. Essentially, it goes away. It essentially doesn't go through, and it behaves like a material.
Yeah.
But it is created out of structures. I mean, designing structures, not by any new material per se. Correct?
Yeah.
Right. Thank you. And so, Jen, you have a question.
Yes. In the later part of your talk, you were talking about that increasing the number of resonators should decrease the energy being damped. Sorry, the increase the energy being damped, but there will be a certain limit. After that, increasing the resonator also is not changing that particular number. So why is that so? What factors are exactly limiting there?
So right now, like we are like I'm optimizing. I'm doing study to optimize like what maximum number of resonator can be used, what minimum number of resonator can be used to achieve maximum reduction. So this is the like still I'm studying about optimization of this number of resonators.
Okay. All right, Sujan, that's all we have. So thank you very much, Kamal. Let's move on to the next talk. I would like Venkatakrishnan to conduct this this particular thing. I will get started, but then the other I would like him to conduct. I need to be away for another activity for something else because of the intense rain that is here. So next talk is by Dr. Hannah Purushothaman. And we have hydrodynamics of channel confined microswimmers. Venkatakrishnan, please conduct this meeting.
Yes, Professor.
Yeah, thank you.
Thank you.
You have also the poster on all the presentations, so you have the speaker details, Venkatakrishnan.
Yes, Professor.
Yeah. Thank you.
Thank you, Professor.
Hello, sir. Is the screen visible?
Yes,
yeah, I think, shall I start then?
Yes, please.
Okay, thank you. So, good evening all. I'm Ahana. I'm a postdoctoral research from the Department of Chemical Engineering, IIT Madras. And today I'll be talking about the research work that I have done during my PhD. And this work was done under the guidance of Dr. Sumesh P. Tampi from Chemical Engineering Department. And the topic for the talk is hydrodynamics of channel confined microtumors. And I know many of you may not be familiar with these words, microtumors or hydrodynamics. So I'll first start discussing what are microtumors. Microswimmers. Hello. Okay, so microswimmers are biological or artificial self-propelling particles of microscopic size. And examples of microswimmers are like bacteria or chlamydomonas, which we see in biological systems. And we know that these microswimmers can swim in the fluid. Depending on the mechanism that these microswimmers uses while they are swimming in the fluid, microswimmers are broadly classified into three. They are pushers, pullers, and neutral swimmers. Pushers are organisms like E. coli bacteria, which generate thrust behind their body using these flagellas, and it swims through the fluid by taking the fluid from the side and then ejecting the fluid through front and back. So it generates a flow field, which is similar to what is shown in this picture here. On the other hand, pullers are organisms like chlamydomonas. It generates thrust in front of their body using these cilia, and it swims through the fluid by taking the fluid from front and back and then ejecting the fluid through side. So the flow field that a puller generates would look similar to what is shown here in this picture. So the flow fields of a pusher and a puller microswimmer looks very similar to a force dipole flow field, with the thrust and drag force acting as the two monopoles. Whereas neutral swimmers are artificial swimmers like Janus swimmers that generates a sourcing kind of flow field. So we can see that all these micro swimmers can disturb the flow field when they are swimming in the fluid. Therefore, it can interact with other swimmers or particles which are present in the surrounding fluid. And these type of micro swimmers are seen in many biological and artificial systems. Though the dynamics of this micro swimmers in unbounded fluid is well studied, the effect of confinement on the dynamics of this micro swimmers is not well understood. And in natural systems, we often see these micro swimmers in confined environments. So these are some of the videos which shows the importance of understanding the effect of confinement on the dynamics of this micro swimmers. So in the first video, what we are seeing is an experimental setup where this E. coli bacteria, which is this green thing, is swimming inside this microfluidic channel. And this width of the channel decreases gradually from the entrance to the exit. So the bacteria enters the channel, which is having a wider width. Therefore, it swims near one of the channel walls. And with decrease in the confinement width, the bacteria starts swimming through the center of the channel. So change in the dynamics from this wall swimming to axial swimming is a consequence of this confinement. So here we are saying that the confinement has changed the dynamics of this particular bacteria here. And in the second video, we see that this suspension of bacteria, which is confined inside the circular geometry. And because of the circular geometry, this bacteria starts forming this collective motion, which is a circular pattern. So the circular confinement has resulted in a collective motion for this suspension of bacteria. And in this experimental study, also when they try to reproduce this experimental result using simulation, they had to consider this hydrodynamic interaction between the bacteria to exactly reproduce this experimental result. So this also shows the importance of understanding the hydrodynamic interaction between the microswimmers in such situations. And in the last video, which is a common biological system, where we are seeing this spermatozoa cells which are swimming inside female reproductive tract. And in these systems, we know that this individual cells are swimming along with other spermatozoa cells, and also it is swimming inside this confined geometry and complex geometry. And in these biological systems, it is important to understand the dynamics of this spermatozoa cells because it is important for fertilization treatment and all. So in all these videos, what we are seeing is that confinement can change the dynamics of this microswimmers, ranging from single swimmer to collective motion of microswimmers. So in order to understand such systems, we need to understand the effect of confinement on the dynamics of single swimmer as well as on the suspension of swimmers. And in case of suspension of swimmers, we need to understand the hydrodynamic interaction between the pair of microsymers as well as the interaction between the microsymer and the other passive particle in the system. So with that aim, we have formulated different model problems for my PhD thesis. And the problems were, first one is to study hydrodynamics of single microsymer in confinement, and then to understand the hydrodynamic collision between microsymer and passive particle. And third one was to look at the hydrodynamic collision between pair of microsymers. So for today's talk, I'll briefly discuss this first problem, which is microsymer in confinement. And this I'll discuss in detail the second problem, which is hydrodynamic collision between microsymer and the passive particle. So we do numerical simulation, and in the simulation, we model the fluid using Navier-Stokes equation and continuity equation. And the microsymers are modeled using a model called Schirmer model, which is an envelope model developed by Blake in 1971. In this model, the motion of cilia or flagella on the surface of this organism are approximated using a surface velocity given by this expression here. It's an infinite series where each term would correspond to a singularity of Stokes equation. And for our study, we consider only the first two modes of the series, which correspond to B1 and B2 modes. And B1 mode gives a source in dipole flow field, and the velocity field decays as order 1 by r cubed. And B2 mode generates a forced dipole flow field with velocity field decaying as order 1 by r square. And beta is another important parameter, which is defined as the ratio of B2 to B1. This parameter is used to model different types of symbers in simulation. If we choose a value of beta, which is greater than 0, we can generate the flow fields of a polar microsymer. If we use a value of beta, which is less than 0, then we can get the flow fields of a Pusher microsymer. If beta is equal to 0, then the flow fields would be that of a neutral symbol. So here I have shown the corresponding flow field for these three types of microsymers. In all these three figures, this white circle represents the microsymer with this red arrow showing its orientation, and this white curve represents the flow field that it generates, and the color represents the strength of the flow field. So this one corresponds to puller, and this is for neutral simmer, and this is for pusher. So if we compare this with the actual flow fields of actual organisms, for example, here I've shown this flow field of a chlamydomonas. This is from experiment. If we compare this flow field with that of a puller simmer, we can see that this direction of fluid motion is same in these two cases. Similarly, this flow field of an E. coli bacteria, which is shown here in this right-hand side picture, which is similar to what we get from simulations for a pusher simmer. So this shows that a Schirmer model is a reasonably good model for these type of organisms. And we perform numerical simulations using Lattice-Boltzmann method. So I'll move on to the first part of the work, which was where we have looked at the hydrodynamics of single microsymer in confinement. And when we studied that, we observed 4 different types of behavior for a single microsymer in confinement, which are represented using a phase diagram here. And the first behavior that we observed is sliding along the wall, which is represented using this red color region here. And as you see in this video here, the sliding along the wall is observed in the case of strong pushers and strong pullers. That is for large negative betas and large positive betas. There, if you look at this video, you can see that a puller swimmer would go to the nearest wall and it starts moving along the wall. This is called sliding along the wall behavior. And the second behavior is periodic swimming along the channel center, that you can see in the case of weak pullers. That is shown in this picture here. Weak puller would bounce back from the nearest wall and it oscillates at the channel center. And the third behavior is periodic swimming near the wall. That is again observed for certain pushers and pullers. In that case, we see that the pusher or puller would go to the nearest wall and it oscillates near the wall before it continues to move near the wall. And the last type of behavior is periodic swimming spanning the channel width. That is observed in the case of neutral swimmers and pushers. As you are seeing in these two videos, a neutral swimmer or a pusher would go to the wall and it escapes from the wall and then would go to the other wall, and it continues to move between these two walls. And this is called the periodic swimming spanning the channel width. So here we see that strong pullers and strong pushers are always trapped and slide near one of the walls, and neutral swimmers and weak pushers show an oscillatory trajectory. So now we'll move on to the second part, where we have looked at the hydrodynamic collision between a microswimmer and a passive particle in confinement. So basically, we wanted to see how different the dynamics of a microswimmer would be if it is in presence of a passive particle. So a passive particle would refer to a particle which cannot move by its own, unlike microswimmer, which is an active particle. So this is the schematic that shows the system that we have considered for our study. And here you can see this red circle, which is the initial location of the microswimmer, and this blue one is the initial location of the passive particle. So the passive particle is initially located slightly ahead of the swimmer and at a distance b from the swimmer center. And we know that the microswimmer generates fluid flows, and the passive particle is present in that flow. Therefore, the passive particle will respond to the swimmer flow field, and at some point in time, these two can come very close to each other, and then it can move away from each other. So the trajectory of the passive particle is represented using this dashed blue curve here, and with P dash being its final location. And the trajectory of swimmer is represented using this dashed red curve, and S prime represents the final location of the microswimmer. And the schematic on the right hand side shows the different type of hydrodynamic interactions that we see in the system. So mainly there are three types of hydrodynamic interactions. First one is between the swimmer and the passive particle, and the second one is between the swimmer and the two walls of the channel, and the third one is between the passive particle and the two walls of the channel. So this is a system with three body interaction, and the dynamics of the swimmer and the passive particle will be determined by the translational and rotational velocities of these particles, which are given by these equations, where u and v are the translational velocity and omega is the rotational velocity of the particle. Okay. So now what we are seeing here is the flow field that a puller microswimmer generates when it is hydrodynamically colliding with this passive particle. So here, this red circle is the microswimmer and the white circle represents the passive particle. So this colored arrows represents the flow field that it generates during motion. And here we are seeing that this puller microswimmer enters from the left side of the domain, and the passive particle is attracted towards the microswimmer. And after the closest approach, the microswimmer escapes from the passive particle and it moves away from the passive particle. So the flow field, if we look at, we can see that it is continuously changing, and it is different from the flow field that a single microswimmer would generate in the unbounded fluid, which I have shown on the right hand side. So this entire process is called the hydrodynamic collision process. So now we'll look at this flow fields in detail. So in all these pictures, this red circle is the microswimmer and the blue circle is the passive particle. And again, this colored arrows represents the flow field that it generates. And here this thick blue arrow shows the orientation of this microswimmer. And this first row corresponds to the hydrodynamic collision that is happening in the bulk fluid. And the second row corresponds to that in the confinement. And also we can see here that this hydrodynamic collision process is now divided into 3 stages: stage 1, stage 2, and stage 3. So stage 1 corresponds to an early stage when the microsimmer and the passive particle are far apart. In that case, the flow field of a microsimmer looks very similar to a force dipole flow field, where we see this top-down and front-back symmetry in the flow field. And in stage 2, we see this passive particle has come closer to the microsimmer. Therefore, this symmetry in the flow field is broken. And also, the microsimmer has now rotated in the anti-clockwise direction. So the flow fields are also rotated. And in stage 3, what we are seeing is that microsimmer has now moved very far from the passive particle. So it regained that symmetry in the flow field. But the flow fields are still rotated. So the difference between the flow fields in confinement and bulk fluid is that in confinement, we see that these streamlines which are closed in comparison to this open streamlines in the bulk fluid. And also the strength of this flow fields are weaker in confinement compared to bulk fluid. So these two main differences can lead to a different dynamics of microsimmer and passive particle in channel confinement compared to an unbounded fluid. So here we see that the hydrodynamic collision process is divided into 3 stages, and flow field in confinement is different from that in bulk fluid. So now we'll look at the trajectories in bulk fluid as well as in the confinement, which are the results of this flow field that it has in bulk and confinement. So in these 2 pictures, the trajectory of microsimmer are represented using this dash curve, and the trajectories of passive particles are represented using this solid curve. And different colors are used to show different stages. So the blue one corresponds to stage 1, and red one is stage 2, and the green one represents the stage 3. So we'll look at the trajectory in the bulk fluid first. Here in stage 1, we see that the microsimmer moves mostly in straight line, because in stage 1, the microsimmer trajectory is unaffected by the presence of the passive particle because they are far apart. Whereas this passive particle responds to the microsimmer flow field and it gets attracted towards the microsimmer. And this stage corresponds to a weak hydrodynamic interaction between the microsimmer and the passive particle. And during stage 2, the passive particle is repelled away from the microsimmer and it moves mostly in the y direction and towards the bottom wall. And during that stage, the microsimmer moves from a position which was behind the passive particle to a position which is in front of the passive particle. And in stage 3, microsimmer again moves away from the passive particle, whereas the passive particle is attracted towards the microsimmer during the stage. So the stage 1 and stage 3 corresponds to a weak hydrodynamic interaction, and the stage 2 corresponds to a strong hydrodynamic interaction between the microsimmer and the passive particle. So now we'll compare this flow fields with that in the confinement. The trajectories with that in the confinement. So if you look at this figure, where I've shown the trajectories in bulk and confinement, we can see that this trajectory of microsimmer in bulk and confinement are very similar, whereas the trajectory of passive particle is different in bulk and confinement. In bulk, we see that this open triangle trajectory for this passive particle, whereas it becomes this self-crossing closed loop in confinement. And also in confinement, we see a net displacement for these passive particles towards the channel wall. And this can be calculated, and it is represented as delta y. It is calculated as the difference between the initial and final positions of passive particle, which are yi and yf. So this is an important parameter.
You have about 5 minutes or so, yeah.
Okay. So this is an important parameter, and it finds applications like particle separation or particle deposition. And there are different parameters that can affect this quantity. And here I'll discuss one such quantity, which is the nature of the simmer. So we can vary the nature of the simmer by changing the parameter beta. So a negative beta corresponds to a pusher simmer and a positive beta corresponds to a puller simmer. This plot here shows delta y as a function of beta. So here for strong pushers, we see that delta y is negative, and with increase in beta, this delta y also increases, and we see positive delta y for pullers, neutral simmers as well as for pullers, and the maximum delta y is observed for the strongest pullers. So yeah. So here we see that stage 1 and 3 corresponds to weak hydrodynamic interaction, and stage 2 corresponds to strong hydrodynamic interaction. And trajectory of passive particle in bulk and confinement are different, and passive particle experience a net displacement in y direction in confinement. So then we wanted to look at the origin of this geometry of the trajectory. So for that, we have used 3 different methods. So this is done to isolate the different hydrodynamic interactions that I discussed in the beginning. So first method is point particle approach, where we approximate this passive particle to be a point particle. So if you look at this flow field, which is the flow field generated by this microsimmer here, and in this method, point particle approach, we have the passive particle, which is like a point particle. So the velocity of this passive particle is given by the flow field generated by the microsimmer at the location of the passive particle. And the flow field of the and the velocity of the microsimmer would be just its self-propulsion velocity u0. And the corresponding trajectories are shown here in this picture, which are the solid line. Again, this red curve corresponds to the trajectory of microsimmer, and the blue ones are that of the passive particle. So this solid curve, if you look at the microsimmer, you can see it moves in straight line. And this passive particle, it shows this open triangle trajectory with this attraction, repulsion, and attraction phases, showing 3 different stages. So this shows that this trajectory of passive particle is a consequence of the flow field that the microsimmer generates. And the second method is the analytical approach, where we account for the finite size of the passive particle. So we correct the velocity of the passive particle with the help of facts and flow, which accounts for this finite size. And also the since it is finite size particle, it will generate disturbance flows, and therefore the velocity of the microsimmer will be changed, and it is corrected by calculating the disturbance velocity that the passive particle generates at the location of the microsimmer, which is this. And the angular velocity is just the curl of this disturbance velocity. So the corresponding trajectories are shown using this dotted curve here. And if you look at the trajectory of microsimmer, it initially moves in straight line, and then it rotates towards the passive particle, and again it moves again away from the passive particle. So this curved trajectory that we are seeing for the microsimmer is purely because of the disturbance flow field generated by this passive particle here. So the third method, which is the numerical method simulation, where we use the 3D lattice Boltzmann simulation, and that accounts for all type of hydrodynamic interactions. So from that, we see that this passive particle has this open triangle trajectory, and the microsimmer trajectory is similar to what we observed from the analytical approach. So these results show that the trajectory of passive particle is a consequence of the flow field that the microsimmer generates, and also the trajectory of microsimmer is a resultant from the disturbance flow field generated by the passive particle. So we see similar trajectories when the passive particle is collided with the pusher simmer and neutral simmer. For the case of pusher, the trajectories are very similar, but the direction of motion of passive particle is opposite to what we see in the case of puller. Here we see first a repulsion and then attraction, and the repulsion for the passive particle. So for pusher and puller, we see 3 stages, whereas for neutral simmers, we see only 2 stages in the hydrodynamic collision. That is mainly because the neutral simmer has a force dipole flow field, whereas for pusher and puller, we have force dipole flow field. So trajectory of passive particle is determined by the flow field generated by the microtumor, and trajectory of microtumor is determined by the disturbance flow field generated by the passive particle. And puller and pusher have 3 stages, and neutral tumor has only 2 stages. So to conclude, collision process is divided into 3 stages. First and last stage correspond to weak hydrodynamic interaction, and second one corresponds to strong interaction. And 3-stage process results in an open triangle trajectory for the passive particle in bulk and a loop-like trajectory in confinement. In microchannel, passive particles undergoes a net displacement towards the microchannel walls, and strong pullers cause the largest displacement towards the channel walls compared to other types of tumors. So, yeah, that's it. Thank you.
Thank you, Ahana.
Yeah, I'll take. Yeah.
You have a few minutes for I think 1 or 2 questions.
Yeah.
There is one question in the chat box. Probably you may look at it.
Yeah, sure.
And
in many cases, these biological tumors use charge to flow in a fluid. They use electrostatic interactions. How different would be the trajectories and the hydrodynamic interactions in a charged species versus uncharged species? Okay, so yeah, I was not very familiar with fact. So I don't know, I'm not sure. I've not come across any cases where biological tumors use charge to flow a fluid. But this is like a simulation where we are trying to model actual biological systems. So whatever be the mechanisms that these biological tumors uses, we can classify them into pushers, pullers, and neutral tumors, and we should be able to understand it based on these trajectories that I've discussed.
okay.
So yeah, can you give an example where we see this biological tumors which uses this charge to flow?
Pallab, you have any examples you may share with Dr. Hannah?
Hi, Anna. I'm not sure what exactly would be the example, but there are many bacteria. For example, probably E. coli must have been many viruses. They have bacteria. They have flagellae, and they are made of proteins. They use as charge different functional groups in the protein, probably amine and all.
Okay, I think even in that case also we would be able to we should be able to classify them under pusher, puller, or neutral tumor, right? By looking at the flow field that is generated.
Yeah, obviously, but there would be,
yeah.
I'm like, my idea would be like there would be minute changes or maybe drastic changes if you apply.
Yeah. So these are like simple models that we use to simulate the actual biological system. So we may not be very closer to the real system. So there will be some factors which are missing in simulation. So biological systems are really complex systems. So we are modeling those systems by taking simple models, right? So yeah.
Yeah, yes, indeed. Sure. Thank you.
Yeah, thank you, Hannah. In slide number 8, when you showed this video, right, you had this particle colliding, and afterwards they separate. What I notice is the white one swimmer, right?
So the white one is the passive particle.
Excuse me.
So the white one is passive particle, and this red one is the swimmer.
Swimmer. Okay. So this once the force field is created, the white is not moving. I'm just surprised to see why white is not moving behind the red.
Okay. So the white thing is the passive particle. So it will move only when it is under that flow field. So once the microtumor has moved far from the passive particle, the flow field would be like stationary, like there won't be any flows.
That's for a few seconds probably, but then afterwards it would take the same direction, I would expect, right? one would expect that the white would also take the same direction as that of the red. Is that right?
No, the white is moving only because it is seeing that flow field that microsimmer generates. So once the microsimmer has moved very far from the passive particle, the passive particle no longer sees that flow field, right? So it will be seeing zero flow field around it. In that case, it will not move because it's the passive particle, right? It needs external force to move.
Okay, but would it move in reality or not? Maybe after a while, once the field is created, to pull it along with it, it should be moving it, right? I'm seeing the many arrows are aligned to push it.
These
forward.
arrows. Yeah. So if you look at the color of the arrows, the strength is maximum near this swimmer. This blue arrows actually shows weaker flow field. So these are actually zero velocities.
Oh, okay. Okay. Yeah. Thank you, Ahana. I think we should go to the next speaker now. Thank you. Thank you for a wonderful presentation. Yeah. So next speaker is Saurav Kar. I think he's going to talk about polyhedral metallo-boron clusters in material science. So Saurav, you want to share the screen and start your presentation. Thank you.
Thank you, sir. First of all, many thanks for giving me the opportunity to present in this platform. So today I'm going to So today I'm going to talk about polyhedral metal-boron cluster, about their synthesis, structure, bonding, and application. So first of all, what is boron? Boron is a metalloid found in nature as various oxide, usually as boron +3. They have two types of naturally occurring isotope, 11B and 10B. So like carbon also, boron has several type of compounds. It comes like such as boron hydrides, organoboron, inorganoboron. It has a wide range of applications starting from hydroboration in hydrogen storage, in BNCT, borosilicate glass formation, metal borides, and so on. So during last 110 years, there are many milestones in polyhedral boron chemistry. In the year of 1912, Alpha Stock began the chemistry of boron. Later on, Slinger, Williams, Wade, Lipscomb, Brown, Hoffman, Mingus, James, and many other pioneers has developed this field in a pleasing fashion. So now take a look at the history of boron cluster. In the year of 1959, Hawthorne and co-worker first isolated the first ever closoborate dianion, that is B10H10 2-, and later on the rest of the boron cluster starting from B6H6, B8H8, B9H9, B10, B11, and B12 were isolated and characterized by Lipscomb, Hawthorne, Mutatis, and others. But the structure of B7H7, which is square bipyramid, this one is synthesized in very poor yield. The yield is 0.0008%, and it was partially characterized by 11 nmr due to the low yield and other difficulty. So this is maybe due to the instability of B7H7, which is proposed by theory. So in this connection, recently we have came out with a new synthetic methodology where group 4 Cp2 metal chloride precursor is treated with lithium borohydride, which led to the formation of metal borohydride intermediate, and further treatment of BH3THF, which is a monoboron, under thermolytic condition. It led to the formation of a metalloboron where the B3 ring, triangular ring, is supported by Cp2 metal BH4 unit from the two sides. So this Cp2 metal BH4 unit, we can consider them as a wing to stabilize this B3. So we thought about, after getting this B3 unit, we thought about why not higher boron cage. So we have varied the reaction condition to get higher Boron case, and the same reaction was carried out under reflux condition and for longer time, which led to the isolation of two more compounds. Here you can see the NMR of these two compounds. One compound has 8 boron peaks in the 11B NMR spectra, whereas the another one compound has 5 boron spectra, 5 boron peak. So we have characterized this 11, the compound which has 11 B peaks with other spectroscopic details such as mass, IR, and finally with X-ray crystallographic study. The X-ray crystallographic study shows that this compound has a B7 core, which the B7 is a pentagonal pyramidal core, pentagonal bipyramidal core, which is supported by the Cp2M BH4 fragment from both sides. The hafnium analog of the same also isolated. So how this B7 core is stabilized by this zirconium borohydride fragment, Cp2 metal borohydride fragment. So to get into inside this the bonding and structure of this molecule, we have carried out theoretical calculation in collaboration with Professor L.A. Soang from Brown University. So they have carried out ADNDP analysis of B7H7 parent. So the orbital of B7H7 parent is presented here from A to E, and the our metal guarded B7, that is from F to N. So if you have a close look in this orbital diagram, you can see that the parent B7H7 has 7 sigma 2 center 2 electron BH bond, and at the same time the our metal guarded B7 also have 7 sigma 2 center 2 electron bond, whereas the parent B7H7 has 5 4 center 2 electron bond. In the case of our metal guarded B7H7, there are 3 4 center 2 electron bond. So instead of this 3 4 section 2 electron bond, in case of our metal guarded B7H7, there are 2 extra 5 center 2 electron bond. So where metal is participated in the orbital contribution. In addition, there are 4 more 3 center 2 electron BH zirconium bond and 2 more B metal BB bond 3 center 2 electron. So these are giving extra electron or extra trivialities to the B7H7. So this is maybe the reason behind the isolation of our metal guarded B7H7. So now we will take a look at the another compound which has 5 boron peak. So in the NMR with equal intensity ratio. So the X-ray analysis study and mass analysis and all other spectroscopy data reveal that this compound is nothing else B8H8 stabilized by Cp2 zirconium borohydride. So the B8 core has dodecahedral geometry. In the, as I mentioned earlier, in the year of 1975, Mutatis et al. Already isolated dodecahedron structure. But what new in our case? So they have isolated the solid state structure, but their solution state structure differ from their solid state structure. So later on, many theoretical and experimental study was carried out, and many studies the solution state structure was reported as square entry prism or bicapped trigonal prism. Recently, Bernadette et al. Reported that B8H82- does not exist in solution state. It is B8H9-. So the solution state structure of B8H82- was mystery for a long time. So we are able to, for the first time, we are able to isolate B8H82- in as a dodecahedron core, both in solid and solution state. The mass and the NMR spectra nicely collaborate with this dodecahedron core. So this time we have collaborated with Professor James from ISC Bangalore to take a look into the insight into the bonding and the structure and the reason behind the stability. The same like B7 core, here also the Cp2 metal BH4 fragment is giving the electron density to the B7H7. So it's also observed in the NDP analysis, and they have performed in addition NOCV plot or EDA NOCV analysis, where they have shown that from metal fragment, the electron is flowing towards the B8 core. So that delta E1 and delta E2 from these two orbital, you can see the contribution is more, where the metal is flowing from, electron is flowing from metal fragment towards the B8 core. Whereas the back donation from the B8 core towards the metal is very less. The contribution you can see 4%, 3%, 2%, and 2%. So the more electron is flowing towards the B8 core from the metal fragment. So that is the reason behind the stability of this type of cluster. So now moving on to the higher vertex, more higher vertex cluster. Till date, no cluster or isolated beyond icosahedron. So what may be the reason? Although Lipscomb and Slater has proposed the structure of higher vertex clusters like 13 vertex docosahedron, 14 vertex bicapped square and bicapped hexagonal and antiprism, 15 vertex henicosahedron, icosahedron, 16 vertex. Two types of 16 vertex structure. One is truncated tetracap truncated tetrahedron, and another one is square crown square. But the theoretic energy calculation has revealed that there is a icosahedron sink. So due to the presence of this sink, the higher vertex boron cluster is not isolated till date. So to solve this problem, James has came out with a nice idea, where he has shown that, utilizing isolopan analogy, that this BH vertex can be substituted by metal fragment. Actually, BH, actually, if you go back, you can see that this all higher nuclearity cluster had higher degree vertices. Like this 14 vertex has 6 connected vertex. This 15 vertex has 1, 2, 3 higher degree vertex, whereas 16 vertex has 4 higher degree vertex. So that's why this higher degree vertex boron was not able to occupy due to their low orbital diffusibility. Whereas metal has more diffuse orbital, and if metal fragment is isolable or isoelectronic with the BH fragment, this metal can be located in these higher degree vertices. So and indeed, Hawthorne et al. Have isolated 13 and 14 vertex metallocarbon utilizing this proposal. So where they have utilized reduced the icosahedron carborane followed by capitation with cobalt CP fragment. So they have isolated 13 vertex as well as 14 vertex metallocarbon. Later on, Joyce, Hosmane, and Alan Wells has isolated many more 13 and 14 vertex metallocarbon and 15 vertex also. Very recently, Joyce and co-worker have reported for the first time 16 vertex metallocarbon in Nature Communication, where they have shown that the structure differ from that of the proposed by Lipscomb and Seiler. This one does not have all deltahedral phase. Instead, it has one rhombohedral phase. And you can see in all these metallocarbon, the higher vertex are mostly occupied by metal, like this ruthenium, here also ruthenium, cobalt. All the 6 connected vertex are located mostly by metal. So that's maybe the reason behind their stability and isolation. So what's about carborane? Very recent, in the year of 2003, Alan Wells has isolated for the first time 13 vertex carborane, utilizing the same reduction and capitation methodology. But this 13 vertex structure, has a geometry of henicosahedron, which is different from that of proposed by Lipscomb and Schweiler. If you look at this structure, unlike docosahedron, this structure has one rhombohedral phase, and due to the presence of this rhombohedral phase, these 2 vertex become 4 connected vertex, and carbon is easily sitting on this 4 connected vertex, and they are able to isolate for the first time 13 vertex carborane. And later on, the Welch and G group have isolated 13 and 14 vertex carborane. And very recently, the Joy group has came out with a 15 and 16 vertex carborane also. So they have utilized the same reduction and capitation methodology. Here, boron fragment has been used as a capitation reagent. And here, the silane group attached to the carbon has acted as the driving force for the isolation of this higher vertex cluster. So this 16 vertex cluster core is proposed by Lipscomb, and they have got the same core, which is square, crown, and square. There is two square in antiposition. In between one crown is there, so it is known as a square crown square structure. So what about metalloboron? So we are working in this field for a long time, and as James proposed that group 9 metals are perfect candidate for the isolation of higher vertex cluster due to their electronic support and their size, atomic size. So we have choose group 9 atom cobalt and utilize the methodology proposed by Fellner, where sequester metal chloride is treated with borohydride, which led to the formation of sequester metal chloride borohydride, and followed by salt elimination and hydrogen elimination, it led to the formation of metalloboron. So utilizing this methodology, we have treated sequester cobalt chloride dimer with lithium borohydride and followed by pyrolysis with BH3THF, led to the isolation of one 16 vertex cluster, cobalt cluster, along with few more cluster. But unfortunately, this 16 vertex cluster is not say single cage cluster, it is a fused cluster, where one icosahedron, one tetrahedron, and one bicapped square antiprism are fused by triangular faces, by 3 triangular faces. And this cluster nicely obey Mingo's fusion formalism and James Emanuel rule. The calculation is shown here. So after that, we move to the rhodium system, where indeed we are able to isolate utilizing the same methodology proposed by Fellner for the monoboron pyrolysis method with metal chloride. We are able to isolate the first ever 15 and 16 vertex metalloboron. The 16 vertex metalloboron has 15 vertex metalloboron has the structure of icosahedron, which is proposed by Lipscomb, and the 16 vertex one has the structure of tetracap truncated tetrahedron. So here all the 16 vertex cluster is shown. This one is the 16 vertex metalloboron, 16 vertex metallocarboron, and 16 vertex carboron. So these are the 16 vertex cluster, which is the highest nuclearity known till date. You can see in the case of the metallocarboron and metalloboron, the highest vertex are occupied by rhodium center and here ruthenium center. So which satisfy the James proposal also. So except the metalloboron, other two metallocarboron and carboron has 17 skeleton electron pair. So these two can be considered as a closure bonding topology, whereas this metalloboron has 16 skeleton electron pair. So this can be considered as a isoclosure bonding topology. So very recently, in this week only, we are working with sequester titanium Cl3 system, and we got on 16 vertex nidometalloboron. And 2-3 days back, we got this structure also, and the other characterization of this nido cluster is on the way. So hope we will get full characterization of this compound very soon. And now moving from polyhedral metalloboron to the classical one. So we all know that boron do not form the flat 6-member B6 ring such as B6H6 like carbon, benzene. Instead, they form polyhedral geometry. So B6H6 will form a octahedral geometry, usually they used to form. But Baldry-Ram and co-worker theoretically predicted that aromatic planar B6H6 minus, the benzene analog, can be isolated by the reduction using lithium, using cis lithium atom. But unfortunately, till date, no success is achieved. On the other hand, theoretical calculation also proposed that, like metal pi complex, like metal CH6H6, like this, or metal P6, arsenic 5, or arsenic 6, like that also boron can be isolated. But there also no success is achieved as a nido arrangement. That is due to the minimum computational for most of the metal. So very recently, James has also proposed that by using early group transition metal, these B6H6 can be isolated. And we have, based on that, we have carried out the reaction of Cp star vanadium Cl3 under thermal with lithium borohydride, followed by treatment with BSC THF under thermal high temperature thermolytic condition, which led to the formation of two types of B6H6 ring with lot of hydrogen support, along with one vanadium B2 B4 vanadium to B4 metalloboron. So here you can see this B6H6 is almost planar. The planar one I have shown here, almost planar, and the how this B6H6 got stability through the 4-3 bridging hydrogen. The neutron diffraction study to finally locate this hydrogen is going on in ANSTO. Hope we will get the final structure very soon. And very recently, we also got one result from titanium system also, where B6H6 ring is present, planar ring, in the like this same thing. And the spectroscopic and spectroscopic characteristics is on the way. So now moving on to the application of this boron cluster. So this type boron cluster has been utilized in polymers, luminescence materials, coordination polymer, nonlinear optics, nanoscience, and binary thin films. For example, the ferroboron and B2H6 Fe2CO6, which is a butterfly core, have been investigated and employed as a volatile precursor for the deposition of iron boride films.
Saurav, just to tell you that you have 4 minutes or so.
Yes, thank you. This is the last slide only.
Yeah.
So boride films with average composition Fe56B44. So with this keeping in mind, we are all now studying this solid state reaction with Professor Bonafide in the from USA. So hope there from there also we'll get some result and get some application of our metalloboron cluster. Thank you all. And I want to thank Professor Sundar Gopal Ghosh, who is my PhD supervisor, and all the Ghosh group members, and my co-workers and collaborators, Professor E.D. James from ISC Bangalore, Professor L.S. Wang from Brown University, Dr. Babu Varghese and Dr. P.K. Sugadevi Atfarajan for their X-ray support, and the funding agency DSTCP and CSR, and finally IIT Madras for their financial support. And thank you.
Very good, very good. Thank you, Saurav.
Thank you.
We are on time. So, any questions from the audience?
Hello, Saurav, am I audible?
Yeah.
Yes, yeah, please go ahead.
Saurav, it's a wonderful presentation. So I want to know that among different polyhedral boron, why carborans or like B12 systems are more stable compared to B8, B7, or B5 or something?
Okay. So here, if you look at this boron cluster diagram I don't have right now the carboron one. Actually, the icosahedron boron is so much stable. If you see the energy, this one is the for icosahedron. So icosahedron is so much stable compared to the higher one and the lower one also. So due to that, this this is more available and easy to make compared to the other carborane.
Is the symmetry is the reason behind this?
Yes, symmetry is also reason. Icosahedral symmetry is the most symmetric one, but most reason is the thermodynamic stability region is the presence of icosahedral and zinc.
Yes.
So if you can see, they have isolated all these carborane in the 1950s, 1960s. So after this, only in 2003 they are able to isolate. After 50 years, they were able to isolate 13 vertex carborane.
Okay.
So that's the reason.
Yeah, so one more thing is that if you go the higher order boron, like more than 12.
Yeah.
So is there any benefit to insert this metal fragment in the femur?
Sorry, metal framework in?
Metal fragment, like this ruthenium fragment or rhodium fragment, whatever you are inserting. Is there any benefit to insert in the femur? Like stability or something?
Yeah. Actually, first, all these higher vertex clusters are isolated by inserting metal fragment only, because this metal fragment is providing more diffuse orbital. And you can see when we are going here, you can see when we are going to the higher vertex, the number of connectivity of the vertex in the cluster is increasing. Here it is 6 connected. Here it is also 6. It has 3 6 connected vertex. It is also has 4 6 connected vertex, 16 vertex. So these higher connected vertex cannot be occupied by boron always. So for that, need more diffuse orbital, and metal has more diffuse orbital. And if they are isolable with boron fragment, metal fragment, so it can be nicely located in that higher vertex.
Then how about the stability of this fragment in compared to normal boron?
Sorry, I did not get your question.
Like what kind of stability is gaining this metalloboron in compared to the higher order boron?
Higher order boron beyond icosahedral tune now not isolated.
Okay,
up to 12 only isolated. So in case of metalloboron and metallocarboron, you can see that up to 16 is isolated.
Okay. So the 12 is the limit for the boron.
Yeah.
Okay, thank you.
Any other questions? Any questions? Okay, so Saurav, I have a quick clarification. These are difficult. I see that complex structures nicely done. How stable are they? I mean, one is how stable are they? Second one is what is the size of them? For example, if you take the larger vertices that you showed, what are their typical dimensions like? How big are these clusters?
Larger in terms of
dimensions. Okay. Vertices, one vertex to another vertex. If you just measure them, how big are they diametrically? You may.
Okay. Actually, I did not measure that. That is a nice idea.
For example, B9 to B11 or B12 to B10 and the below structure.
Yeah.
How big are they in terms of the dimension? Typically,
with that here you can see this type of cluster, all boron are connected, right?
Yeah.
Means they're making a like a aromatic 3-dimensional and aromatic structure kind of.
Yeah.
So that boron this
Sora, what Venkat would like to know, I think you're going a different direction. Probably okay. He need the space inside.
Yes.
Like in fullerene and all those molecules, like whether you can trap something, do some catalytic work, or maybe he has some special interest. I know that. So you need to see it crystallographically. What is it?
Yeah, I need to see it, sir. I never measure it.
So the dimension is one thing, and the stability wise, how stable are they? They're okay at room temperature. You can
means you cannot keep for long days, but under atmosphere you can keep it.
You can keep them. Okay. So the crystals, whatever you grow, are quite stable. Looks like.
Yeah, quite stable.
Quite stable. Okay. So usually the data are collected at a lower temperature, no? Or you have a special method to do in case if it.
In most of the case, we used to collect in normal room temperature, but in few cases, we have to collect in low temperature. For example, I will show you one.
Mounting could also be tricky.
Yeah. This B8H8 was we got crystal after 1 year, and for mounting it takes 6 months because it's immediately we are taking out from solution. The crystal is opaquing.
That's a common observation.
Yeah. So this one is this one has makes so many problems. Other ones are okay.
It's okay. So what is your experience like when you keep a metal? They are okay to keep them at normal optimum conditions, but when you don't have metal, they are quite unstable. Or how do you comment on these things? If you want to comment.
So it is for in our case, it's usually best to always keep under argon atmosphere.
Both with metal, without metal. How do you compare them in terms of stability?
I think with metal, in this crystal, it's not a good idea to keep outside.
Okay. So in both the cases, when you might have to be careful in terms of storing them and collecting the data. So that is quite challenging and tough thing to do. Yeah, good. I see nice results are coming up. Thank you very much, Saurav. Are there any questions? Okay, there are no questions. Please join me in thanking Saurav. Thank you, Saurav.
Thank you, sir. Thank you very much.
So I think with this, we are done for today, this month's meeting. I think thank you very much for all the participants. Thank you very much for all the speakers who have been part of this session. Yeah. Thank you very much. Bye.
Thanks, Venkat. Thank you.
Thank you so much. Bye. Suraj, shall I close? Shall I end the meeting?
Yes, sir, we can end it.
Okay, yeah, thank you.